Welcome to the Society for Marine Mammalogy's Editor Select Seminar series. Seminars that are given in this series are papers that are selected by the Board of Editors, chosen to be considered for a seminar. We record the presentations and they are then posted on the Society's YouTube site. I should let you know that if you have questions, they should go in the Q&A rather than in the chat, although we should monitor the chat as well. And if we do get questions there, hopefully we'll be able to ask them as well if time is available. So my name is Daryl Boness. I'm the editor-in-chief of Marine Mammal Science, and I have the pleasure today of introducing our speaker, who is Dr. Brenna Frasier. Brenna received a master's degree and PhD degree from Trent University in Canada, and is currently, and I was Brenna very currently, curator of zoology. Her work over the past 20-some years has focused on using ancient DNA as a tool to address questions about evolution and conservation, and also about species management. Brenna's latest work is focused on North Atlantic right whales and beluga whales. He's also studied and published on the horses of Sable Island, which for those of you who know about sort of Eastern Atlantic or the Western Atlantic and Nova Scotia, Sable Island horses are pretty popular animals. She's also worked on walruses and has studied several bat species. Besides her research and publishing her research, she's enthusiastic about facilitating and communicating science to the broad public. They explain part of the reason why she's curator of zoology. This has led to her involvement in a number of outreach-based projects, including the development of marine mammal and forensic science camps for teenagers, supporting a local forest school, and writing field guides to assist in traditional ecological knowledge studies on marine mammals. So today, as you can see from Brenna's introductory slide, she'll be talking to us about Eastern North Atlantic right whales, and will be presenting some of her genetic analyses. As you can see, she has a long list of co-authors. I just need to give them to you since they're right there in front of you. And her paper, her talk is based on the paper with a similar title, Genetic Examination of Historical North Atlantic Right Whale Bone Specimens from the Eastern North Atlantic, Insights into Species History, Transoceanic Population Structure, and Genetic Diversity. The paper is currently in early view. It hasn't been assigned to an issue yet, but her paper is open access, so it is available to all, not just those who have memberships in the society. So with that, I will turn it over to Brenna and look forward to her.
Thanks, Daryl. And thanks so much for the invite. I'm always really happy to talk about our right whale research and the way that we're investigating the history of the species. So first, I'd like to acknowledge all of my co-authors. We're a pretty large multidisciplinary team of scientists, archaeologists, and historians. And this work is a result of many hours of lab work, but also decades of archaeological field work and research. So I thought I'd start off with just a little bit of background on the North Atlantic right whale. So the right whale is a large baleen whale species reaching about 50 tons and 50 feet in length. The species is critically endangered with about 400 individuals remaining, and its primary range is from along the eastern coast of the United States and Canada within a pretty urban coastal environment. And the status of the species is largely a result of a pretty complex mix of factors. So this includes the species history with a long history of whaling, but also contemporary threats within its habitats. And then again, on top of that, some intrinsic challenges as well. So some of the extrinsic factors affecting the species are because its right whales suffer considerably from anthropogenic mortality from vessel strikes and entanglement with fishing gear. So as an example of this, 86% of right whales in the population show signs of entanglement, some sort of entanglement event at some point in their lifetime. Now, an added layer of complexity here, particularly in terms of how we try to manage and monitor the species, is the fact that the ocean environment is changing. And so while right whales used to extensively use the Bay of Fundy as a prime area for feeding in the summer, with changing ocean conditions, they're starting to move their locations along with the scientists that are studying them, and shift their habitat use to the Gulf of St. Lawrence in the summers. So you can see here in the distribution, right whales spend their winters in a calving ground, at least females mostly do, along the coast of Florida, Georgia, South Carolina, and then they move northward in the spring and summer. And so they've shifted their habitat into the Gulf, which has posed some additional challenges for the species more recently. So in addition to these extrinsic factors, they also have some intrinsic factors that are posing some challenges and appear to be limiting recovery. So first, they have a low reproductive rate. It's about 1/3 of what is thought to be possible for the species. They also have a really highly variable calving rate, calving intervals. So for example, a single female might give birth every 3 years as they're kind of capable of, but the next calving interval might be 15 and the next might be 20. And there's variability within females, but then there's also huge variability across females. And we think that has to do with something intrinsically that is limiting reproduction. And so in addition to all this, they also have low levels of genetic diversity. So this low diversity has commonly been attributed to kind of genetic loss over time, and potentially because of erosion of alleles or bottleneck events that have resulted from the reduction of the population due to whaling events. So together, all these factors have really left us wondering if recovery for the North Atlantic right whale is possible, and also whether we can actually alter those factors that we do have control of effectively enough to save the species. So in addition to contemporary anthropogenic mortality, it's widely recognized that the status of the species with so few individuals remaining is largely a result of almost 1,000 years of commercial whaling activities that occurred all over the North Atlantic. So right whales were once, shown in gray here, once distributed across the North Atlantic. And then whaling for the species kind of began in earnest in about the 11th century in the Bay of Biscay off the coast of France and Spain by Basque whalers. And then whaling activities in the North Atlantic followed kind of a counterclockwise movement. So the hunt expanded northwards in the mid-14th century towards the coast of Ireland and England, and then by the 15th and 17th centuries to waters off Iceland, Norway, Greenland, and the shores of eastern Canada. I'll talk more about eastern Canada in a minute. I've highlighted that in yellow. By the 17th to 18th or 17th to 20th centuries, right whales were also hunted along the shores of New England. And then in the 19th and to mid 20th century, they were sporadically hunted all over the North Atlantic until they got international protection in 1935. So I've spent a considerable amount of my career chasing whale bones in search of evidence of these whaling activities, and more specifically looking for bones from early right whales in hopes of really being able to quantify how exactly whaling has impacted the species. So are the low levels of diversity we see a result of whaling activities, or has the species been struggling for far longer than we've thought, perhaps as a result of prior climatic events or something like that. So I mentioned that bass traveled to the coast of Canada to hunt right whales in the 16th to 17th century. And so it's been suggested that a huge proponent or a huge kind of aspect of the loss of numbers for the North Atlantic right whale was a result of this particular phase or period of whaling, with an estimated 40,000 whales hunted. And so we spent several years along the coasts of Quebec and Labrador finding old Basque whaling sites and collecting bones to try to find evidence of right whale past. And we found a lot of bones, and we collected hundreds of samples. But the majority, in the end, when we conducted genetic analyses on these bones, the majority of these bones were bowhead whale with a single North Atlantic right whale bone. And so it looks like, at least here in Atlantic Canada, this 16th to 17th century Basque whaling, which was thought to have been a pretty huge impact on the species, wasn't actually focused on right whales. It was focused on the bowhead whale. And so it didn't have as big of an impact as we had thought previously. And the second thing that we found from this right whale bone was that when we looked at the genetic profile and compared it to the contemporary population, it looked just like, genetically, it looked just like an individual within the population today. And so it looked like genetic diversity, at least in the 16th to 17th centuries, were very similar to the levels of diversity that we actually witness in the species today. So they were pretty low before much commercial whaling started for the species. And I had the added pride of publishing a paper with a sample size of 1. So we know that right whales were once present on both sides of the North Atlantic, and that they were exploited first in the eastern North Atlantic and subsequently around the ocean and into the western North Atlantic. Today, the species only remains in that area I showed earlier in the western North Atlantic, within a pretty small portion of what once was its former range. And so a major question regarding the historical status of the species has been how many populations existed prior to whaling. And really, there's been very little data available on which to make any sort of strong interpretation. And the question has been largely unanswered. But understanding this past population structure and the interactions among right whales could give us insight into whether there's potential for individuals in the western North Atlantic to potentially reoccupy areas of the eastern North Atlantic and go back to those historical habitat areas. And this is especially, there's potential for this as the climate changes and oceanic conditions are changing for the species. And we're already witnessing that with these shifts into the Gulf of St. Lawrence. And so there's been historically 3 hypotheses that go along with this. The first one is that there was historically a single population. So under this idea, animals from these regions may have shared habitats, but also had some temporal isolation. So I've numbered 1 there, the one population kind of map idea. And so, for example, right whales may have had separate calving grounds in the eastern and western North Atlantic, but then shared offshore summer feeding areas around Iceland and Cape Farewell. Under this scenario, the idea would be that knowledge or kind of cultural memory of these areas may have been lost due to the extirpation of whales that use them. And therefore, those areas that might have had to do with those matrilineal lineages have since been lost. And therefore, these areas haven't been reoccupied. The second hypothesis would be that there are 2 populations. And so 1 in the western and 1 in the eastern North Atlantic. And based on historical observations and known whaling records, the eastern population may have spent winters along the coast of Northwest Africa to the Bay of Biscay and summers in the waters that range between Iceland, the North Cape of Norway, and the British Isles. And then the western population would have wintered between Florida and New England, and then summered in the coastal waters ranging between the Gulf of Maine and Labrador. This hypothesis, I would say this has been kind of the most widely accepted idea about the hypothesis of population structure for right whales in the North Atlantic. And it's been based on 2 primary lines of evidence. The first is that the eastern North Atlantic region has not been repopulated by individuals from the western North Atlantic. The second is that contemporary and historical observations of migration patterns on both sides suggest more of a north-south migration history as opposed to kind of transoceanic. And then further, each of these areas appear to have kind of distinctive calving areas in the south as well. There was a third hypothesis, and that was that there were three potential populations, with one considered more of an offshore or mid-Atlantic population that could have spent winters in the waters off of the Azores or Bermuda, and then summers around Cape Farewell. And really, to date, there's not a huge amount of evidence to support this hypothesis, so I won't kind of talk too much about that. So as part of a much larger scale study that's looking at 1,200 years of marine mammal use around the North Atlantic, we recently genetically identified a whole array of marine mammal species. And among these were 24 North Atlantic right whale bones. So these samples originate from 10 different sites and range in age from the 4th to the 20th century. What's neat about these is that they really give us kind of a rare representation of the extirpated eastern North Atlantic right whale. And so it's an opportunity to really try to better understand the history of the species. So not only does this give us another look at historical levels of genetic diversity, but it also allows us to examine the characteristics of right whales in the eastern North Atlantic. And so within this larger scale project, we've now analyzed over 300 bones from 45 sites, and we found 17 species to date. And so the paper that I'm talking about today that Daryl mentioned, that's published in Marine Mammal Science, this is the examination of the right whale bones that we identified within this much larger sample set. And so this is just a map showing the distribution of the right whale in the western North Atlantic, and then little circles with stars in them is where we have our 24 right whale samples from that were a part of this study. And again, these are from 300 to about 1912. This is just an example of some of these specimens and what they look like. What's exciting from an archaeological perspective is that a lot of the time these sorts of specimens can be identified as definitely being cetacean or marine mammal by their structure by archaeologists, but we can't, from the morphological features, identify the species. And so genetic analysis really adds a nice layer of information about animal use across sites, within sites, and what species were there. And you can see here I've highlighted two that actually show signs of some sort of working. The one on the top there has definitely been used and fashioned into a shape. It's a very nice artifact. It's a lovely piece. And then the one on the lower left there has some cut or hack marks on it. So these are bones that have been used to varying degrees. So here's a breakdown of the samples identified as right whale based on where the samples came from. We found 10 specimens in Iceland, 5 from the Faroe Islands, and 7 from Scotland. So these were from Shetlands, Orkney, and Hebrides. And below this, what I've done is highlighted the age of the sites or the context of the sample within the sites where these specimens were found. And so one thing I should note is, so Iceland, we have samples that originate from sites kind of 800 and beyond. There's a wide spread of samples in terms of age from Scotland. And then the Faroe Islands have one peak in the center there. And I would say that's largely a factor of the fact that we have one site in the Faroe Islands and several bones from one site. So that's a bit of an artifact in the interpretation of that graph. So for this study, we compared ancient DNA, looking at mitochondrial DNA and nuclear DNA from these historical specimens to contemporary genetic specimens and profiles that have been collected previously. So over the past 40 years, there's been continued fieldwork and photo identification and collection of samples from the North Atlantic right whale. And we now have about 80% of the species genetically profiled looking at nuclear and mitochondrial DNA. So we have a huge sample set to which we can compare our historic data to. So our aim was really to assess the genetic characteristics of the historical eastern North Atlantic right whale and test hypotheses about population structure within the species. We extracted DNA from about 300 milligrams of bone shavings that were collected from each bone. And then in the slides that follow, what I'd like to do is discuss the analysis and results that we did in terms of looking at species ID and also looking at diversity and population structure. And we did this using, again, both mitochondrial DNA and in particular the mitochondrial control region. And for nuclear DNA, we ended up developing 15 locus microsatellite profiles. So species ID was confirmed by constructing a Bayesian inference phylogenetic tree. We looked at 445 base pairs of a fragment of the cytochrome B gene, and we were able to get complete sequences in order to determine haplotypes for 16 out of the 24 samples. And for those that came short, what we ended up doing was looking at those. So those were under 400 base pairs. And so what we ended up doing was doing visual comparison of those sequences to known species. And then we also did blast engine searches to identify the species with a criteria of 100% coverage and 95% maximum identity. And so this was what we did for about 8 of the 24 samples. And so for the assessment of diversity and population structure, we compared historical samples and data previously collected from the contemporary population. And again, that was about 80% of the species. And for mitochondrial DNA, we did 3 things. So we constructed a median joining network, which is a really great way to visualize how your haplotypes and your mitochondrial DNA, that data falls out. We looked at diversity indices, and then we also conducted a standard analysis of molecular variances using FST and phi-ST. For the nuclear data, we ended up having 8 samples that were good enough in terms of preservation to obtain DNA profiles. So we assessed differentiation using FST and GST, and then we also looked at heterozygosity as another kind of insight into changes in genetic diversity. So here's the median joining network. So this is a nice way to visualize kind of all of the known mitochondrial control region haplotypes that we have to date from the Western and Eastern North Atlantic. So what this is a visual representation of each of the haplotypes shown as circles and how closely related they are to each other by lines across between circles. And so any differences between these haplotypes are shown as dashes on each of the lines. And the larger the circles are here, the greater the proportion of samples that are comprised that have that haplotype. And so where you also see white dots, that really denotes any sort of inferred haplotypes or those that are missing in the linkages. So on the top in the light blue, you can see that the haplotypes that exist in the contemporary population, these are haplotypes A through G, in blue circles, there's many missing haplotypes. So it's kind of a holy network. And then if you look at the middle image in dark blue, you'll see that in the historical samples, we found many of these missing haplotypes. So we kind of filled in that network. And I've also included all other right whale haplotypes for this region. So we have a single haplotype identified from that 16th century bone from Labrador that I mentioned. And then there's one other that we know of that has been developed, and this is from an 18th century right whale baleen stomacher. It's a decorative clothing piece that's stiffened with baleen. And we don't know the origin of this, but there's one published haplotype associated with that I've included here as well and called it textile. And when you examine the network, what is immediately evident then is that all the new haplotypes that we find fall closely within that star-like structure of that network without any sort of clear structuring or patterning that's really associated with geographical distribution or origin. And it's also notable that when observed alone, the contemporary network at the top exhibits several of those missing intermediate haplotypes amongst those found within the population today. And then obviously that historical network does not. So when we look at differentiation, what we did was quantified using a standard AMOVA approach. We were concerned that there's large differences between the sample sizes of the two data sets that we would be comparing. So I think we have something like 575 right whales, contemporary right whales sampled as a part of our data set. And we have a pretty small number of historic samples. So to get around this, what we ended up doing was conducting 1,000 simulations that were comparing the differentiation between the Western and Eastern samples to that calculated just among the Western samples if they were subdivided in the same manner. So we would randomly sample the same number of Western samples and compare it to the remaining Western group in the same way that we'd be doing the comparison of historic to contemporary. So in gray is shown the differentiation distribution just within the Western North Atlantic as a result of these simulations. And then in black, we show the differentiation between the West and the Eastern North Atlantic. And you can see that there's more differentiation when looking between the West and Eastern North Atlantic than if you were just looking within the Western North Atlantic alone. We did something very similar for the nuclear DNA. So here is differentiation, showing differentiation FST using 1,000 iterations. And so in gray is differentiation within just the Western North Atlantic. And then in black, we show that are actually quite different, with differentiation being greater between Western and Eastern than just within the Western samples. Looking at our mitochondrial DNA diversity indices, while nucleotide diversity is actually relatively similar between the two groups, there's much greater haplotype diversity in the historical sample set than in the contemporary sample set. And so when we look back to the nucleotide diversity, these similar values reflect the fact that the actual haplotypes in both sample sets are actually very similar to each other. They usually differ by only one single nucleotide from each other, and this results in similar degrees of expected differences between each of the groups, or within each of the groups. And so we suggest that the difference in the haplotype diversity in comparison to this, the haplotype diversity values are different, and we suggest that this reflects a couple of things. The first is our sampling strategy. So the historical samples were, of course, collected over a very long time period, or not collected, but originate from a very long time period, greater than 1,000 years. And it's from a historically larger population containing a larger number of mitochondrial DNA haplotypes, while the contemporary sample set is certainly collected from a much smaller time period and a smaller extant population. So you see this big difference in those haplotype diversity values. The second reason for this is likely the fact that the species has undergone pretty severe population reduction as a result of whaling activities. So we've lost diversity, and we see lower level of diversity in the contemporary population. For the assessment of nuclear diversity, in the end, we were able to get profiles from 8 samples. We looked at 15 loci, and in the end, we had 4 complete profiles and 4 that were incomplete. So those that were incomplete, we were only able to successfully obtain 7 to 13 loci. And we found 55 alleles in total looking across all 8 samples. So this included 2 new alleles that have not been identified in the contemporary population. And most of those 55 alleles, those that remain, are present in the population with pretty common frequencies between 0.002 and 0.775. And we found 1 rare allele, which was present with a frequency of less than 0.005. So what you might expect if you had two populations that were significantly different than each other is that you would find in one population many alleles that are rare in another. That might be the expectation. So levels of heterozygosity are often used as a signal of high levels of diversity and also can be examined to detect changes in diversity. So to assess whether this observed heterozygosity differs between the Western and Eastern samples, we compared the observed microsatellite heterozygosity of the 8 Eastern profiles to that of the contemporary population. And again, we did 1,000 random subsets of 8 Western samples so that we're not swamping our data with that huge sample size on one side of the kind of comparison. So in gray, you can see the distribution of observed heterozygosity values for 1,000 iterations. And in black, you can see the observed heterozygosity of the historic samples. And this value falls right within the center of the distribution of values calculated for the contemporary Western population. So this really suggests that there's no significant difference in heterozygosity between these two sample groupings. So we've been able to examine historical diversity in the species both on a temporal scale and across the North Atlantic. Both mitochondrial and nuclear data indicate some degree of genetic loss over time. And until now, we haven't really had a lot of data available to look at in terms of historical diversity. My previous work on that single right whale bone from the 16th century Labrador suggested that the historical population had similar levels of diversity to the population today. We did find a new matriline at the time looking at that single bone, and we suggested that that was perhaps because whaling in northern regions had resulted in the loss of a matriline of animals, North Atlantic right whales, that use that area. And this is based on evidence that North Atlantic right whales do show significant matrilineal structuring in these important summer feeding areas. So our recent data set, we've again found that there's been a loss of many mitochondrial DNA haplotypes, but a smaller signature of nuclear DNA differences. And so again, we suggest that the loss of many haplotypes for the species may be indicative of a higher degree of historical population structuring that was once reflected in mitochondrial DNA, that is only reflected in mitochondrial DNA and not nuclear DNA. And so this might be expected if the species exhibited some degree of mixture in mating areas, but site fidelity in northern regions in summer feeding areas. Indeed, all our samples are from northern regions, and they would have represented summer feeding areas. So this interpretation fits well with observed historical and contemporary characteristics of other Eubalaena species globally as well. So we set out to examine whether right whales in the North Atlantic historically comprised one or two populations. Certainly, we're limited in the conclusions we can make because we don't have much of a historical sample set from the Western North Atlantic. Ideally, it would be lovely to compare historic samples from the same time period from both sides of the ocean and do that direct comparison. Our data sets are challenged because they differ not only spatially, but also temporally. So there's limits to what conclusions we can draw. But we do suggest that this comparison is a little less tenuous because of the fact that when we did look at that single 16th century right whale sample from the West, it was genetically very similar to the contemporary population. So that gives some support that we're not doing a tenuous comparison between the West and the East. Coming back to the question of how many right whale populations may have existed prior to whaling, we suggest that the one population hypothesis seems unlikely for several reasons. First, historical observations show evidence of temporal and spatial isolation of right whales in the Western and Eastern North Atlantic, with some degree of latitudinal seasonal distribution. Second, contemporary research has found that right whales in the Western North Atlantic show fidelity to their habitat in the region with no evidence of reoccupation of the eastern North Atlantic. And then finally, the identification in our data set of a relatively large number of those new mitochondrial DNA haplotypes, as well as some degree of nuclear differentiation between the West and East, is not concordant with a single interbreeding population. So it would be expected that if the two populations of right whales existed that were completely genetically isolated over many thousands of years, we might have this lovely clear pattern of genetic differentiation between the two. But it's never that simple. The challenge with the North Atlantic right whale is that, and species like it, is that they have it takes a long time, an incredibly long amount of time for such differentiation to occur, especially since individuals are so long-lived and have such long generation times. It takes much longer. So it's possible that the population could be genetically isolated, but not show a clear genetic signal of such differentiation if they were connected relatively recently in the past. We see this, for example, in bowhead whales, where they have been connected fairly recently, and populations in the Pacific and the Atlantic look relatively similar genetically. So we identified some differentiation of both nuclear DNA and mitochondrial DNA across the North Atlantic, with slightly greater differentiation in mitochondrial DNA. And there are shared microsatellite alleles and mitochondrial DNA haplotypes also across the regions. So there's not complete reproductive isolation between the two sides of the ocean. What seems most plausible, at least this is what we have suggested, is something in between these kind of maybe textbook, non-realistic, clear-cut versions of a 1 or a 2 population scenario. So our results show that specimens found in the eastern North Atlantic were genetically similar to those in the West, but with a little bit of differentiation. And so it's really difficult to tease apart whether this sort of observed differentiation is a result of genetic shifts due to population reductions from centuries of whaling, whether it's due to a signature of historical population structure across the ocean basin, or is it some combination of both factors? So although we don't know from what the difference results, we can confirm that the animals from both sides of the ocean basin had at least some degree of gene flow, and enough to make them genetically similar. So we suggest that the observed slightly greater degree of population mitochondrial DNA differentiation is a result of maternally directed site fidelity to summer feeding grounds and or calving grounds on each side of the North Atlantic. So one of the things that we discussed in our paper was whether we might gain insights into the possibility of right whale recolonization of the eastern North Atlantic. I've said we found a small degree of differentiation between the West and the East. And so this may suggest that that repopulation in the North Atlantic of the East is not limited by genetic constraints. They weren't so different that it's kind of an impossible idea, but it's more likely that reproduction may be limited by the loss, or sorry, not reproduction, repopulation may be limited by the loss of that sort of cultural knowledge of the existence of these areas in the whales that have survived following commercial whaling. And so it shouldn't be ignored that there's a possibility that new kind of ecological and behavioral barriers to repopulation also exist, and that this could be affecting the Western population and may be preventing reoccupation. It may be the case that contemporary conditions have created a habitat that the whales are avoiding, or it's also possible that the species has not yet recovered to an adequate population size with enough kind of internal pressures to warrant emigration occurring to reoccupy these habitats. So to kind of wrap up here, the complicated history of the North Atlantic right whale, with a sort of very uncertain demography and centuries of commercial exploitation has left us with a challenge to really define indices of species recovery. The species has suffered a long and complicated history of whaling and is currently struggling to survive in the 21st century. This recent paper in Marine Mammal Science really illustrates that interpretation of historical population structure is also not always straightforward, and sometimes the details are impossible to disentangle. So we suggest that management strategies be diverse and collaborative, evaluating not only the current condition of the species, including those intrinsic factors that I mentioned earlier, but also the status of the species in the past. I think this is particularly important in light of potential habitat use shifts with climatic changes to come. And finally, this study really does highlight the scientific value and the great potential resource of historic specimens such as those that we use for providing really important insights into current species population genetics, and to support the development of more effective conservation and management strategies as well. So for our next steps for this work, we're now conducting whole genome sequencing of several North Atlantic right whales. And part of the kind of goals of this work are to examine population history, including demographic changes over time, and try to get a better sense of kind of what's happened in the past for this species, what was pre-exploitation population size, and with kind of newer, more advanced techniques looking at whole genome data, I think we're going to get some really interesting insights into the background of this species. So thank you very much, and I'll take questions.
Thank you, Brenna. I see we do have some questions in the Q&A, so I'll turn it over to Teresa, who is going to manage the Q&A.
Okay.
Yeah, I'll go ahead and read the questions we have so far. And like Daryl said, please continue posting in questions and we'll read them out. So we have our first question from Sonia wondering, could I ask what poachers hunt the whales for? In the historical whaling, what were they trying to get?
Yep. So right whales have one thing interesting about right whales in comparison to a lot of the other baleen whales is that they have incredibly long and flexible baleen. And so certainly for centuries, baleen was a big attractant. It was kind of one of our first plastics. It's really very flexible and could be used for all sorts of functions, for umbrellas and corsets and all sorts of things. They also have an incredibly thick blubber layer, and so they were hunted for their oil, which you can get from the blubber, but later on, well, for a lot of whaling periods, you could also boil the bones. They hold a substantial amount of oil in their bones, so whalers would extract oil from the blubber and from the bones, and then that was used to light the streets of the world, and among other things, as lubricants for machinery and all sorts of things. And to some extent for food, but not hugely.
Great, thanks. Okay, our next one. Is there some other reason for why that one sample was found in the west? Perhaps it traveled there through whaling vessels from the east. Perhaps I missed that part. My apologies if you already explained.
No, that's great. Actually, what's really interesting about that question is that when I was drafting as a PhD student, when I was drafting that paper, a couple of my co-authors, there were so many other possibilities, like maybe right whales were hunted elsewhere, or maybe they were hunted first and the right whale bones are underneath the bowhead whale bones. And to be honest, I think there's a lot of different potential strategies or possibilities for how the single right whale bone got there. But I think the most realistic is that the whale was hunted there, and there wouldn't have been a huge reason to transport bones. They're big, they're heavy. It's a big humerus, and so I think in the end we had a wide swath of samples from shores and underwater. We had divers, and so we really looked quite thoroughly to try to find right whale bones and didn't. And so we're going on 300 to 1 bowhead to right whale.
Okay, from Kara or Kara, sorry if I'm mispronouncing that. Given your research and conclusions, can you speak to the sighting of the right whale calf in the Canary Islands a few years ago?
Yeah, there's been a few sightings of right whales around old habitat areas. So there, in the last few decades, there's been sightings of right whales off the Azores, the Canaries, off Norway, off Iceland. And what's interesting is in several cases, it's been known individuals from the Western North Atlantic. So we have some wandering whales from our Western North Atlantic population. And so far, I think the question is always initially, is it a remaining Eastern North Atlantic right whale? And for the most part, it appears like they're animals from the Western North Atlantic that are moving around. Sometimes they don't look as healthy as maybe they could, but I think for the most part, I think Mogul was seen off Iceland and was seen later back on the coast of the US and Canada. So yeah.
No, we actually had another question asking about slalom and mogul. You've been sighted near Iceland. So yeah, that was another question from someone else.
Yeah.
From Lee, we have, has there been evidence of sound pollution being significant to North Atlantic right whale migration?
There are a lot of people kind of monitoring noise in the water, certainly. I don't know that I'm trying to think of what examples I know from right whales of kind of evidence of an impact, a clear demonstrated impact of the noise. I know that during just following 9/11, I believe there was a huge increase in right whale vocalizations off the eastern shores of the United States. And I think that is one paper, I hope I'm not wrong about that, but I think that's one published paper showing that certainly their vocalizations increased significantly in quieter waters. And so to me, that is an impact of ocean noise for sure.
Great. From Mark Alexander, we have, thanks for the talk, very exciting. I have two questions. First, and this might be related to what we were just talking about, I recall there was an individual sighted in Iceland a couple of years ago. Has that individual been identified, or is the sex known? So I guess we'll ask that one first. Is that the other two you were talking about, or is that a different animal?
I'm pretty sure that's Mogul, and I actually don't know if Mogul is male or female. I'm not sure. Sorry. Yeah.
So related to that, we have, would it be possible to molecularly sex any of the historical samples, or is the DNA too fragmented?
I think we could, and to be honest, that's something we have a technique that we've developed actually to sex sloughed sperm whale skin that works on really, really tiny fragments of DNA, and we just haven't tried it on the bones yet. So I think there is potential to do that. And if not with something like qPCR, I think if I can get a long enough fragment to do microsatellites, then we should be able to sex the bones. It just needs some time and money.
Always time and money. From Erica Hill, we have, were historic whaling practices for bowhead and right whales identical, or do they require maybe different types of technology or strategies?
Physiologically, right whales and bowhead whales are very similar. Bowheads are a bit bigger. They have a longer baleen and thicker blubber layer. Anecdotally, what I have heard is that bowheads are the right whales can be pretty rough. Like they thrash a lot and they're kind of tough to work with, and bowheads are a little more calm, but also more skitterish. This is absolutely not a scientific answer. But just in terms of anecdotal comments I've heard from people who work with each of the species, the temperaments of the two are quite different. And so I don't know if that would affect whalers' choice in terms of, do you want a real thrashing whale or something that is not quite as, but I think certainly that techniques would vary for bowheads if you were kind of further north in more icy waters and that sort of thing. The bowheads are more ice associated, and so the seasons would be different for right whales and bowheads. In theory, you would have bowheads that come down more in the winter, whereas right whales also have gone further south in the winter. So they shouldn't really overlap much in terms of whaling activities. And that actually brought up some interesting questions about seasonality of basque whaling along the coast of Canada, because they had at one point what they talked about as a spring, like a spring whale and a fall whale. And so it's not known whether, the spring whale was bowhead whales heading back north into the Arctic, and the fall whale was potentially right whales that were in the area. Did I just get that backwards? Yeah. But yeah, so whether it's two different seasons for two different species doing different things in their migrations. So.
Great. From Jennifer, have you considered working with indigenous people, to be a source for collaboration for acquiring samples of ancient bones?
Yeah, it all depends on the project. Certainly for bowhead whales, we have, maybe not with indigenous communities, but we've got samples from people from the Geological Survey that have been doing work up in the Arctic. We have in the past worked with indigenous communities to get samples from contemporary species that we work with. So I did some work on walrus, and we certainly were able to get to work with them to get samples from them. In terms of where right whales are distributed, at least in Canada and the United States, there's not a lot of sites I can think of off the top of my head where we know there could be right whale bones that we could access and work with indigenous communities to find. But I think there could certainly be a lot of potential there. There's so much community knowledge on history and where those specimens might be. I think it would be such a great collaboration.
Great. We have another question from Lee asking a little bit about what types of solutions are possible. And I'm assuming that's solutions to the population issues and the low numbers of North Atlantic right whales.
I think we need to stop killing them. What we really can control are vessel strikes and entanglements. Right whales are, entanglements a huge one. They're going not only with ghost gear, but live active gear in the water. The right whales are very prone to getting entangled, along with many other species. But I think the more that we can try to prevent interactions of those species with boats and with gear, that's something we can directly affect, if we had the right kind of strategies in place. And I know both the US and Canada are working hard to have different management areas when right whales are present to control and try to mitigate the effects of ships and shipping lanes. Certainly in the Gulf of St. Lawrence, right whales now, years ago, my colleagues worked to shift shipping lanes in the Bay of Fundy so that they don't go through critical right whale habitat. And now we've been posed with a new issue of, pretty major shipping lanes that go through the Gulf of St. Lawrence, and the right whales have now entered the Gulf of St. Lawrence. So one of the biggest issues has been that we spent 30 years maybe in working around the coast of the US and Canada to try to manage these interactions. And now the right whales have shifted. So it's a whole new fishery. It's a whole new group of shipping lanes. And so we really have had to pivot to try to manage what's happening and prevent more deaths.
From Helena, we have, does the similarity in heterozygosity between contemporary and historical populations suggest that the extent of inbreeding was similar?
Not necessarily. I think that heterozygosity and inbreeding are kind of two different. They're a little bit more different than that, and so I'm not super qualified to comment on the extent of inbreeding. I think any work that we've done on inbreeding in North Atlantic right whales in terms of trying to assess whether they are inbred because of these low levels of diversity in a small population has shown that they don't show any kind of strong signatures in inbreeding. But I know there are members in our lab that are looking further into this.
Great. We've got a couple minutes if folks want to keep posting questions. Daryl, I don't know if you have any you want to ask.
Yeah, maybe I'll ask one that's more policy related. And well, I'm curious whether you think the ability to take mitigation measures is easier in Canada than in the U.S., given how long it often takes for changes to occur through the various legal channels.
Honestly, I don't know. I have been impressed. I've been impressed at how fast Canada has reacted to the shift into the Gulf of St. Lawrence. There's almost, it seems like when there's right whale sightings, there's almost immediate kind of events that happen to slow down ships and make areas, make management areas where, the way ships have to behave are different and the way fishermen have to behave is altered. It seems like we've reacted quickly, but in Canada in particular, we've also seen big shifts depending on what government is instated, whether we have kind of liberal or conservative management at the time. And so it really changes by decade and depending on what's going on.
Great, thanks. That's a tough one to answer.
Yes.
So sorry for putting you on the spot on that.
Yeah. No, I'm sure several of my colleagues are in this call and could probably answer better, but yeah.
Okay, do we have anything else in the chat, Teresa, or are we?
So we have one chat question that maybe Lee can clarify, asking, are there economic solutions being discussed to compensate whalers? Now, my understanding is there's not a lot of active whaling going on right now, right? So I don't know, Lee, maybe if you want to clarify that question. In the meantime, I'll go ahead and ask one on the part of the students. We like to ask if you have any general advice for people who are wanting to either pursue a PhD in the area that you pursued or end up in a similar career to yours. Anything that's really been beneficial for you in your path to where you are today.
Volunteering. I think volunteering is an excellent way to get experience if you can afford it and make it happen. I know it's sometimes costly because it's not always free to volunteer. Sometimes you have to pay to get there. But any way that you can volunteer, even to get your foot in the door doing something that maybe doesn't seem directly applicable, but you build trust and confidence with the people you're working with and then get more responsibility and experience. I had a couple opportunities to volunteer as an undergrad. One was doing frog research, but it all affects the way you think about things, so it doesn't have to be directly marine mammal related. Sometimes it's challenging to get into the marine mammal world, but from myself and from those I'm close with that study marine mammals, certainly some of their volunteer and summer research experiences were some of the most valuable for making bonds and deciding whether they do or don't want to go a certain direction. I've got colleagues that would get seasick and realize they couldn't be in a plane doing aerial observations or on a ship for days on end. I think also following leads that excite you. You might start off with a project that is one thing, but just from my experience during my PhD, I started off on a project looking at right whales, and I could have kind of just stuck to that. But I realized there was, from geological surveys, there were all these bowhead whale ear bones from 10,000 years in the Canadian Arctic that were available. And so I kind of don't be afraid to reach out to people and ask for collaboration or ask for ask those questions. It never is a bad idea to kind of seek out those potential side projects that might seem weird, but they can take you places that you have no idea about yet.
So I see we are, unless any other questions have popped up, we're right at 3:59, almost 4 o'clock here. So Do we have a follow-up on that one you had asked about earlier?
We do have a follow-up. I'll
Okay,
go ahead
go
and
ahead.
read it out. First, they clarified that they were thinking about indigenous populations that might still be doing sort of subsistence whaling, and if you have any thoughts on that. And then also following up, are there alternatives for communities or corporations that rely on fishing or vessel use? And I'm assuming this is with vessel strikes and those sorts of issues.
Right. Yeah. So for at least for right whales, there is no indigenous whaling for right whales that occurs. I know with other species like the bowhead or the gray whale, there's a lot of work that goes on to try to strike a balance between kind of maintaining indigenous cultural practices and also managing the species. And so there's certainly, I don't know about kind of monetary compensation or any sort of compensation that way, but certainly there's a lot of dialogue and research that goes into any of the decisions towards a hunt of those more rare and threatened species in particular. In terms of what's been neat to witness in terms of right whales and vessel strikes and entanglements is there's always conflict, but there's also a lot of collaboration. There's a lot of people doing research to try to find and develop fishing gear that is not as likely to entangle whales. I mean, really cool stuff like satellite released lobster pots that are at the bottom with no gear at the top. And I mean, there's really interesting kind of developments. I think 10 years ago, someone was coming up with Viagra rope that was stiff when it was in the water so that right whales would be less likely to get wrapped up in the line. And so there's a lot of kind of research endeavors going into developing better technology. I think there's also a lot of communication and education, both back and forth between the fishing community and scientists and researchers about the activities of the whales and where they are and what they do. And I'm not saying it's all flowers and butterflies. I think there's a lot of conflict that goes on, and these are people's livelihoods that are also at stake, and that's threatening. But in my experience in the right whale community, it's been really positive, some of the dialogue and the communication that goes on.
Great. Thank you so much. And thank you, Lee, for clarifying that question. That was really helpful. I think that's it on our end. So, Daryl, do you have any final words you want to say?
We're at 4 o'clock here, so let me just thank you, Brenna, for a very interesting talk. I'm glad to see your interest in kind of communicating to a broader public. I think that's also extremely important in getting younger people involved and giving them opportunities is critical to have people like you working to do so. So thank you for that.
Thank you.
So I guess with that, we'll come to an end, and we'll see you again next month. Third Thursday of the month is when we have our seminars. Time varies depending on where our speaker is located. So thanks again, Brenna.
Thank you.
Your time.
Thank you. Have a good day. Bye.
Bye-bye, everybody.